Hello, everyone. Myself, Dr. Daisymon. I'm working as an assistant professor in the physiotherapy department, University of Science and Technology, Meghalaya. Today, I'll be discussing a small topic that is blood. So, what blood is? So, as we know, blood is the only liquid connective tissue that is derived from mesoderm. So, what is mesoderm? It is one of the germinal layers during the embryonic development period. So it is we can say blood, it is one of the main circulatory fluid that is present in our body. So the study of blood, it is known as hematology. So we can consider blood as fluid of life, fluid of growth, and fluid of health. Why? Fluid of life because it carries oxygen from lungs to all parts of the body and carbon dioxide from all parts of the body to the lungs. So there is a exchange of oxygen and carbon dioxide uh, as blood being the medium. On the other hand, fluid of growth because blood carries the nutritive substances from our digestive system and hormones from the endocrine system to all the parts of the tissues. Next, why we call it as a fluid of health? Because it protects the body against diseases. It protects our immune system, and also it helps in get rid of unwanted waste products that are being accumulated during various metabolic activities to get excrete out of our body with the help of our excretory organs like kidney, skin, liver, etc. So, so this is one of the images of blood structure. So when we take a sample of blood in a test tube and we centrifuged it and keep it aside for a few minutes, then we can say that then we can see that blood gets separated into different layers. On top there is a yellowish portion, a liquid yellow portion, which is known as plasma, which constitute about nearly 50 to 60% of our blood. Whereas at the bottom, there is the cellular portion, the thick solid portion, which is known as RBCs, the red blood cells, which carries hemoglobin. And in between plasma and RBC, if we notice very carefully, we can see a very thin white layer, which can be identified as WBCs. So, so when we centrifuge a sample of blood, so blood gets separated into different layers like plasma, WBCs, and RBCs. Coming to the composition of blood, blood, as I've just now said, blood is composed of two parts. One is known as the formed element or the cellular element, and the other part is known as plasma. The formed elements constitute about 45% of the blood, which consists of RBCs, WBCs, and platelets, whereas the plasma, which is the liquid portion, yellow liquid portion, which constitute about 55% of our blood. So, here in the image, I have clearly mentioned that platelet constitute about 4.8%, RBCs, which holds the maximum, that is 95.1%, whereas WBC, it is 0.1%. WBCs, as we know, it can be classified into again for the two parts, that is granulocytes and agranulocytes. Depending on it, we have neutrophils, eosinophils, basophils, monocytes, and lymphocytes. The percentages are being mentioned there in the screen. Whereas the plasma, when we look at the plasma, it constitutes about 55% of blood. So the maximum portion of plasma is water content, which constitute about 92%, and the remaining portion of plasma may be electrolytes, protein, waste products, nutrients, and various gases which we get in a traceable amount. So the protein that we find in plasma, it's albumin, globulin, and fibrinogen. So these are the three proteins that we find we find in plasma. And the other substances may be gases like nitrogen, oxygen, carbon dioxide, even urea, uric acid, glucose. All this in a very traceable amount. These are being present in plasma. Coming to the properties of blood, as we know, color, blood is always red in color, but if we look more if you notice blood more precisely, the arterial blood is really more deeper in color. That is, it is scarlet red, whereas the venous blood, it is little bit, you can say purplish red or little bit blackish red because it carries deoxygenated blood. On the other hand, volume, normal adult, uh, normal volume in a normal adult will be 5 liter, where in case of females, it may vary because as we know, female have a uh, little bit less blood compared to male. So it may be 4.5 in case of female. In a newborn baby, the volume of blood is 450 ml. Coming to the pH, it is 7.5, 7.4 or 7.4 to 7.6 also. And if we compare, if we look at the pH, it is, the blood is always in an alkaline. It is in between alkaline state, so slightly alkaline. So the pH would be 7.4 to 7.6. Coming to the specific gravity, if we come to look at the specific gravity of the total blood, it is 1.052 to 1.061. Whereas the specific gravity of blood cells, that is the solid portion, is 1.092 to 1.101, whereas the specific gravity of plasma is 1.022 to 1.026. So the numerical readings may vary to book also, so it doesn't matter. Coming to the viscosity, blood is 5 times more viscous than water. So if you look at the blood, the consistency or the thickness of blood, it is little bit on a thicker side as compared to water. So as because blood contains the solid portions like RBCs and plasma protein. Next we come to properties of blood. So properties of blood. First property is nutritive function. Nutritive substances like glucose, amino acids, lipids, and vitamins derived from the digestive food are absorbed from the gastrointestinal tract and carried by blood to different parts of the body for growth and production of energy. So whatever food we consume, as we know, that get converted, once it gets digested, that get converted into different substances like glucose, amino acids, vitamins, and these substances are being carried to blood. These substances are being transferred to blood to different parts of our body for various activity, for various functions to run, for the proper running of our human body. Next, respiratory function. As we know, blood acts as a medium for exchange of gases like oxygen and carbon dioxide in our alveoli of our lungs. Next, coming to the excretory function. Waste products formed in the tissue during various metabolic activities are removed by blood and carried to excretory organs like kidney, skin, liver, etc. for excretion. So, whenever, as we know, our human body, inside our human body, there are a lot of metabolic activities continuously going on. So during this metabolic activities, a lot of unwanted waste products are also being simultaneously deposited in our body. So those products or those unwanted substances need to be excreted out of our body. So those things, those unwanted substances or that are being deposited during all the metabolic activities like digestion, burning of calories, your urine formation or whatever activities. So those unwanted substances has to be excreted from our body, and blood helps to excrete those substances through kidney, skin, liver, which are the excretory organs in our body. Next become transfer of hormones and enzymes. We know hormones are being maximum of hormones in our body that are being directly that are secreted directly from the ductless glands. So as we know, ductless glands means directly transferred to the blood. So that's why blood is directly helps to transfer these hormones and enzyme to different parts of our body and tissues for proper functioning. Next, we come to regulation of water balance. Water content of the blood is freely interchangeable with interstitial fluid, which helps in regulation of water content of the body. As now, just now I have said, in the composition of blood. Plasma contains, the water content in the plasma is almost 92%. So, the blood indirectly helps in the water balance of our body during dehydration or during uh, mm, some activities where there is lack of water content in our body. So that has been that is maintained by blood. So next we come to the sixth point, that is regulation of acid-base balance. Plasma protein and hemoglobin act as a buffer and helps in regulation of acid-base balance. So blood, we know it is slightly on an alkaline side. So the plasma protein and the hemoglobin content in our blood helps to maintain that acid-base balance, to maintain the blood at a pH of 7.4 to 7.6 level, which is very, very necessary for the proper functioning of our body. Next, regulation of body temperature. Blood is responsible for maintaining the thermoregulatory mechanism in the body, that is the balance between heat loss and heat gain in the body. So the thermoregulatory mechanism, that is the balance between the heat loss and the heat gain in our body, that is being maintained by our thermoregulatory mechanism. So blood helps in maintaining the thermoregulatory mechanism. Next point is the storage function. So water and some important substances like protein, glucose, salt, sodium, potassium are constantly required by the tissue. Blood serves as a ready-made source for the substances during conditions like starvation, fluid loss, electrolyte loss, etc. So, besides absorption, besides transfer of this substances like protein, glucose, amino acids, or excretion of unwanted substance, blood also acts as a reservoir. So it acts as a storage. So storage of some substances like protein, glucose, amino acids in our body. So those substances are being stored, and these are being utilized in our body during activities like during conditions where we are in starvation, or where the person is having fasting, or where there is dehydration in our body or loss of electrolytes in our body. At that time, those storage substances are being acts as a buffer, and they help in the regulation of the function of our human body. Next, defensive mechanism or defensive function. So we can say the WBC are the responsible for the defense function. So the WBC helps in maintaining the immune system of our body to protect the body from various bacteria, viruses. So it is the WBC which helps in protecting our body. So next we come to, more precisely, we come to the point, that is RBC. RBC, which are also known as erythrocytes. So when we look at the morphology, that is how the RBC look, how RBC cell looks under a microscope. So I've shown you the image. It looks like a button shaped. So they are non-nucleated formed element in the blood. So generally they are biconcave, where the central portion is thinner and the periphery is thicker. So they look like somehow buttons. So they have their own diameter, as I have mentioned, it is 7.2 millimeter. The surface area 120 square mu, and volume is 85 to 90 cubic millimeter. So normal value of RBC range from 4 to 5.5 million cubic millimeter of blood. In adult male, it is 5 million cubic millimeter, and in adult female, it is 4.5 million per cubic millimeter. So as we know, the lifespan of RBC is 120 days, or it may be 110 to 120 days. So function of RBC. So main function is transport of oxygen from the lungs to the tissue, as I have mentioned earlier. Transport of carbon dioxide from the tissue to the lungs. Next, buffer action in the blood. They act as a storage of substances, and which are being utilized during emergency conditions like starvation or electrolyte loss of a person is having in dehydration. So blood acts as a buffer action in the buffer action. Next, in blood group determination. So it is through the RBC only, the blood group what the individual have, whether the person belongs from group A, group B, or O plus, O negative. So it is the RBC which determines the blood group of an individual. So next we come to the physiological variation and pathological variation. Physiological variation, that is the variation that you see during normal condition. It may be increase in the number of RBC or it may be decrease in the number of RBC. So physiological variation is quite normal, whereas pathological variation is during condition or during diseases. The fluctuation in the number of RBCs or WBC, which is not healthy and which needs immediate treatment or management. So that comes under pathological. So physiological variation, where there is increase in the RBC, that is age. At birth, the RBC count is 8 to 10 million per cubic millimeter of blood. The count decreases within 10 days after birth due to destruction of RBCs, causing physiological jaundice in some newborn babies. So we have heard that newborn babies always they used to suffer from jaundice. That's quite normal. It's called physiological jaundice. So during birth, the RBC count is from 8 to 10 million per cubic millimeter of blood, quite normal. But within 10 days, it get back into a little bit on a higher side, but within 10 days, it get back into a normal count. So it's quite normal to have an increase in RBC count at the time of birth. Next, sex. We know that male has comparatively more RBC count as compared to female, because female, they undergo menopause, they undergo menstruation, so there is a little bit fluctuation in the RBC count. Whereas high altitude, inhabitants of mountaineers having an increased RBC count of more than 7 millions per cubic, it is due to hypoxia in the high altitude. Hypoxia stimulates kidney to secrete a hormone called erythropoietin. So erythropoietin is a hormone that has been secreted by the kidney. So it helps in the formation of RBCs. So those who are living in the mountains, they have a comparatively more RBC count as compared to those living in plain area. So you can say high altitude people will have a more RBC count than the people living in the plain area. Then during muscular activity, after meal, emotional conditions, stress, in all this condition, the RBC count or can get increased for a few hours, but it's quite normal, because during muscular exercises, we know there are a lot of pumping activities after meal. Automatically, a lot of metabolic activity, our digestion process starts. Emotional conditions, stress, happiness, at that time our RBC count will increase. It's quite normal to have in this condition, in this physiological condition, so that the RBC get increases. Coming to the decrease in RBC count, that is high barometric pressure. That is at high barometric pressure, as in deep sea, where the oxygen tension of the blood is higher, the RBC count decreases. So the divers, in case of divers, we can see the RBC count is quite decreasing in their number, because as they go deep into the sea, the oxygen content there decreases, and there is a lot of oxygen tension in the blood. So the RBC count decreases in case of divers. During sleep, as we know, there is no such activity. So during sleep, the RBC count may decrease, quite normal. But once we get up and get back to our work, so RBC count going to get back to the normals. Pregnancy, the RBC count decreases because of increase in the ECF volume, that is extracellular volume, extracellular fluid volume. Increasing the extracellular fluid volume increases the plasma volume, also resulting in hemodilution. So there is a relative reduction in the RBC count. So in case of pregnancy, so RBC, you can see the RBC count is decreases. So it's not so very difficult, serious condition. It's quite normal because of the increasing ECF volume. So the RBC count gets decreases, but quite normal. Nothing, it's nothing to worry or think during pregnancy. Then we come to the pathological variation. Pathological. So we can see the in pathological variation in polycythemia, respiratory diseases like emphysema, congenital heart diseases, carbon dioxide, chronic carbon monoxide poisoning, poisoning like chemicals like phosphorus and arsenic, repeated mild hemorrhage. So this may be the few conditions, or you can say a few conditions where there may be some pathological variation in the RBC count. And in this condition, there is a need of treatment or management for the patient. Next, this is a slide of slides showing the stages of RBC. So there are 6 stages of RBCs, that is normoblast, pronormoblast, polychromatic erythrocyte, autochromatic erythrocytes, reticulocytes, and erythrocytes. This is the last one is the matured RBC. So before that we have 5 stages. This can be visualized. This is a microscopic image. So that can be very beautifully visualized in a microscope, under microscope. So under microscope, the various stages of RBC look like this initially, and in the last stage only we can see the red color slowly comes as the color of RBC is red, and in the last stage only you can see that button shaped structure coming out. So next we come to WBC, which is also known as leukocytes, which are colorless and nucleated form of elements of blood. So as I have said, the WBC color is colorless. Number, it is less than 4,000 to 11,000 cubic millimeter, which has a size of 18 mu. Shape is irregular. When we talk about WBC, it is generally, we say it is an amoeboid shape. When we look at the microscopic diagrams, images of WBC, we can see the variety shapes of WBC molecules, WBC cells. So nucleus, WBC has a nucleus, whereas RBC don't has a nucleus. They may be WBC may be granular or agranular. Based on it, only we have divided into granular and agranular. And WBC has a lifespan of up to 15 days, or you can say 11 to 15 days. They have a lifespan. So these are the classification, and also this you can see the different images of WBC, how the neutrophil look, eosinophil, basophil, lymphocytes, monocytes. This is a diagram there, image. This is an image of this molecule. Image of the cells of WBCs, different types of WBC, neutrophil, eosinophil, basophil, monocytes and lymphocytes, that is. Next, again, we come to the physiological variation of WBC. Age, WBC count is about 20,000 per cubic millimeter in infants and about 10,000 to 15,000 per cubic millimeter of blood in children. In adult, it ranges between 4,000 to 11,000 per cubic millimeter of blood. So this is the normal range according to age. Sex, slightly more than in female. As I said, male has a more WBC count compared to female. Diurnal variation, minimum in the early morning, maximum in the afternoon. Maximum, you can see the WBC count rises in the afternoon. Exercise, sleep during exercises, as I said, increases. All the activities in our body increases. So WBC count also increases during exercises. Next, we come to sleep. During sleep, the WBC count decreases. Emotional conditions, anxiety, fear, depression, in all those conditions, it increases, WBC increases. Pregnancy, menstruation, parturition, in all those conditions, the WBC count increases in our body, which is quite normal in physiological variation. Next, coming to pathological variation. When we talk about pathological variation, we come to know about different condition diseases. So first is leukocytosis, which is the increase in the total number of WBC count. And we can see leukocytosis in condition like allergy, common cold, tuberculosis, fever, in some infection. So in these conditions only, in this condition, the leukocytosis, there is increase in the total WBC count. In condition, next we come to the condition that is leukemia. We know leukemia is what? Blood cancer, where there is abnormal or uncontrollable increase in the number of WBC. So which is more than, you can say, 10 lakh per cubic millimeter. So in leukemia, it's again a pathological condition, and it is directly it is a case of treatment, immediate treatment. Next, we have leukopenia, which is the decrease in the total WBC count, which occurs in some pathological condition like liver cirrhosis, spleen disorder, liver disorder, in some anemia, typhoid, or some in viral condition like viral infection. In all those conditions, you can see leukopenia, which means there is decrease in the number of the WBC count. The word leuko is WBC, and penia. Penia means what? Decrease. So the word penia means decrease. So there is in all these conditions like cirrhosis, liver cirrhosis, disorder of spleen, typhoid, some infectious fever. In all those conditions, there is a decrease in the WBC count. Next, I have shown you an image of the stages of WBC, which is known as granulopoiesis, or you can say leukopoiesis also. Okay. So the stages of WBC is known as granulopoiesis or leukopoiesis. So we have myeloblast, promyeloblast, promyelocyte, myelocyte, metamyelocyte, band form, and granulocyte. So these are the stages of a WBC. Ultimately, we used to get a granulocyte. Okay, so you can, on the microscope, you can see the structure looking like this. So we have almost 6 stages of WBC to form a mature WBC. Next, last we come to platelets. Platelets or thrombocytes. Platelets are also known as thrombocytes, which are form elements of the blood. So these are small, colorless, non-nucleated, and moderately refractive bodies. So in the image, you can see the platelets these are the white structure which are around the RBCs. These are platelets. So they have an irregular shape. These are colorless, non-nucleated structure. So they have a diameter of 2.5 mu, volume of 7.5 cubic mu. Normal count is between 2 lakhs to 4 lakhs cubic millimeter of blood. And the lifespan of platelets is 8 to 11 days. So in the image, you can see the platelets. Next, we come to the physiological variation in same age. Platelets are less in infant and reaches the normal level at the 3rd month after birth. So the count of platelets during birth, it is less, but once the baby attains 3rd month or the infant attains 3rd month, so it becomes normal. Sex, as I know, as we can say, male will have a more platelet count, or you can say male-female ratio may be also same. High altitude platelets count also increases as we have seen in RBC. After meal, after taking food, platelets count again increases. Quite normal after meal exercises, platelet or emotional condition, and all those conditions, the platelets count will also increase in the number. Next, we come to the pathological variation. We have condition like which is known as thrombocytopenia, which is again known as decrease in platelet count. So the decrease in platelet count is known as thrombocytopenia. So in condition like acute infection, leukemia, chickenpox, smallpox, typhoid, tuberculosis, in all those conditions, you can see a decrease in the platelet count or the number. Next, we have thrombocytosis, which is known as the increase in the platelet count. So it can be during surgical condition, it can be during hemorrhage, rheumatic fever, severe trauma. So in all those conditions, there may be increase in the platelet count. Next, we have thrombocythemia. Thrombocythemia is a condition with persistent and abnormal increase in the platelet count, such as carcinoma or leukemia, chronic leukemia. So, as we know, leukemia or carcinoma, it is related with cancer. So, thrombocythemia is very, very, you can say, little bit on a dangerous side, which immediately needs treatment. So overall, when we look at all the pathological variation or all the pathological condition, these are all related to treatment. Next, we come to the stages of platelets. These are the stages of platelets, so which is known as thrombopoietin or megakaryopoietin. So the different stages of formation of platelets is known as thrombopoietin or megakaryopoietin. So we have the stages like megakaryoblast, megakaryocytes, then we have the platelets precursor extension, and then finally we have the platelets. So these all stages we can see very nicely under microscope. So lastly, we come to the plasma. Right? So plasma, as I said, it is the liquid yellowish portion that you find in when once we centrifuge the test tube and we keep the blood sample, we find that one liquid portion, yellowish in color, on top of the cellular element that is known as the plasma, which contains about 92%, 91 to 92% of water and 8 to 89%, as I explained before, so solid, which may be gases, which may be other unwanted substances, nutrients, etc. I've also mentioned that the plasma contains plasma protein, which are albumin, globulin, and fibrinogen. So coming to the normal values. So the normal values of plasma protein are total is 7.3 gram per deciliter. Serum albumin is 4.7 gram per deciliter. Serum globulin is 2.3 per gram deciliter. Fibrinogen is 0.3 gram per liter per deciliter. And when we look at the overall ratio between albumin and globulin, it is always 2:1. And fibrinogen, as we know, it is the clotting agent. Next, we come to a small portion that is known as serum. So serum is the clear straw liquid fluid that oozes from the blood clot. When the blood is shed or collected in a container, it clots. In this process, the fibrinogen is converted into fibrin and the blood cells are trapped in this fibrin formed cells. In this fibrin forming cells, and they form the blood clot. After 45 minutes, serum oozes out of this blood clot. So serum is what? The plasma minus the fibrinogen. Fibrinogen is the clotting agent. Without fibrinogen, blood doesn't get clot in our body. So serum is what? It is the clear straw liquid fluid that oozes after the blood clot. Okay, that you see after 45 minutes when the fibrinogen gets converted into fibrin. You can see a small white sticky liquid substances that get deposited on the wound. So these are serum. So when we say serum, it is always the plasma minus the fibrinogen content of the blood. Okay. So this is overall small briefing, small topic on the blood, its composition, the structure, how they look, a brief on the stages, various stages, functions in our body. With this, I would wind up my topic today. Thank you.